Hello class, today we're going to talk about our macromolecules. A little bit of this will be some review, but a lot of new things and a lot of just different things to focus on from when we learned about a few of these before. So the first thing we're going to start with is learning which four molecules are our macromolecules, which are carbohydrates, proteins, lipids, and nucleic acid. Another good reminder is if I have it underlined, that's just a good thing that I would love for you guys to be writing along in your notes. If you are taking notes in your notebook, I highly recommend that you do. This is the most important information that I want you to be doing. So here's our first little note is that there is going to be some organic chemistry in here, or OChem for short. I'm going to help you guys learn a little bit about what these different ways to write chemical compounds mean, and we'll work through it together. So our first thing we're going to learn about is carbohydrates. So our definition for a carbohydrate is an organic compound containing long carbon chains with hydroxyl, ketones, and aldehyde groups. Need to remember that our term organic means that we're using carbon, hydrogen, oxygen, and sometimes nitrogen. And also that to make up carbohydrates, we have a bunch of monosaccharides, or this is also a term for a sugar, a simple sugar, but the scientific word we'll be using is a monosaccharide. But also going back to our definition of what a carbohydrate is, it's these organic compounds which are full of carbon, hydrogen, oxygen, and nitrogen with these three different kinds of groups. So we have an aldehyde group, which this R means that it can be any part or any chain of organic molecules, any part. And then we have a double bond. So this is a double bond to an oxygen and then a hydrogen off the other side. Our ketones have one group of one chain of carbon and hydrogen, and then a different one. R prime just is signifying that it's not identical. It can be one chain and then a completely different size and length of chain with a oxygen in the middle, double bonded, and then a hydroxyl is we have our R group and then an OH. So those are for the three different groups that we all have on there. I just wanted to kind of introduce you guys to some of that because we will be seeing it in our next slide. So for example, we can see that we have all these long carbon chains, and we have this hydroxyl group, hydroxyl group, hydroxyl group off the side with a, I think this was an aldehyde. Yep, with an aldehyde on top. So we have our aldehydes, and then we have a good example of how our hydroxyl groups can flip sides or all be on the same side. There's also this difference called an alpha glucose and a beta glucose. I just wanted to introduce you guys to this. So alpha and beta are just showing us telling us which carbon that this OH is going to be on. So alpha meaning it's on opposite sides and beta meaning it's on the same. This simple change, like we can see over here, changes a lot about the way that this glucose molecule can bind to other ones, and then will create different structures. So just some interesting, more detailed chemistry I wanted to expose you guys to, but I'm not going to expect you to remember or learn, but I just wanted to show you. So our next next notes that we're going to write down are carbohydrates can create branched or unbranched structures. So for example, we have a good little picture here where we have a bunch of polysaccharides or carbohydrates as unbranched, nice organized little lines. We can think of unbranched as like our bricks. If we're laying bricks, they would just stack on top of each other, where if we had branched stuff, it would look like a tree. It would look much like this. So trees are a good example of our branched carbohydrates. Our branched carbohydrates are used in energy storage, and some examples are starch and glycogen. And our unbranched ones, our brick laying ones, are used in plants for cell walls and cellulose. So these unbranched are structural, much more structural, and these branched ones are much more energy storage. So that comes back to our functions, our important phrase that our structure determines our function. So if we have our main function is to store and provide energy, and we talked about those branched carbohydrates doing a lot of that storing. And then for our structure and our support is our other function. And so those are when the unbranched and our bricklaying type of carbohydrates come in handy. The next macromolecule we have are proteins. So we've learned about proteins before, but we're gonna learn about them again in a new way. Before we were talking about how they could have been created for life, and now we're going to talk about the importance of their structure and function. So our structure is a complex structure made up of amino acids joined by peptide bonds. We learned that last time is our definition, and then we also defined amino acids as organic molecules containing a carboxyl amino and an R group. So when we look at our carboxyl acid, our amino acid, we have these three parts. We have our amino group on the left, we have our R group on the top, and a carboxyl group on the side with this middle carbon. And then also I think we touched on last time is that when we join many of these amino acids together to get our macromolecule of a protein, we take this hydrogen from one amino acid right here and the hydrogen of another, and we lose this H2O, which is water, to form a bond. So we can still see this double bonded oxygen on this carbon, a nitrogen, and a hydrogen. That is called a dehydration reaction. So we also learned that our body uses 21 different ones, and that peptide bonds form through dehydration reactions. So here are the groups of amino acids which are the most common. We can see that they are grouped in this table by R groups that are electrically charged side chains. So if there's a positive charge in them, positive, I don't see a charge in this one, but I'm sure there's a lot of partial charges. These are full charges. I'm sure there's partial charges in here for this positive, and then our negative charges. They will have a lot of different electrostatic interactions, electrical interactions, because our opposite charges will attract. So if we had an amino acid that had this as an R group right next to an amino acid that had this lysine as an R group, the R groups would want to interact in their own ways. We also have amino acids that have polar and uncharged sides. So we talked about the importance of polarity when we talked about our water and how it can kind of really attract or work with different molecules in specific ways. This last one is hydrophobic. So we have polar, which would uncharged, which would be hydrophilic, and then hydrophobic chains. So this would also act in very unique ways, especially in our cells, which are full of water, and then we have a couple that don't quite fit into any of these three categories. So our protein structures, there are 4 different structures, and the combination of R groups determines our structure. So the different amounts of these and in the different orders that they're in really will change the shape dramatically. So our first structure is our primary structure. So we have this being our amino acid sequence. We can see from this little picture that it's literally just the amino acids in a line and nothing more. Then we have our secondary structure, which indicates the presence of an alpha helix or a beta sheets. So our alpha helix is just a nice coil. It looks like a spring. And our beta sheets are, this is not a great picture. Hopefully we can see some more later, but are lines that kind of form. And then our third one is our tertiary, which has our 3D folding. So our alpha helix and beta plated sheets are still 2D, like we're drawing on paper. And our tertiary comes in when we can fold it maybe like in half, like when we're folding paper, and make it more three-dimensional. And then our tertiary is just multiple, or sorry, our quaternary is just multiple tertiary subunits coming together, like they have over in our picture. So this can change a lot, but it's important to remember. I have a quick little video to maybe visualize it a little better. So you don't need to hear what the video is saying. I really just want us to see the. Yep, so I just wanted us to kind of look through what it might look like. So this is our primary sequence. We have a nice chain of our different amino acids. They all have different names. And then as we, oh, sorry. I want to go back. Sorry. So this is our, primary. Here's our secondary. So this is a good picture of what our alpha helix might look like. It's nice and coiled. In between these little dashed line are hydrogen bonds, which we learned about also when we talked about water and the importance of hydrogen bonding. And then we also have our peptide bonds, which are covalent bonds. Our beta-plated sheets look more like this, that kind of line up side by side, whereas our coils will circle. So this is our tertiary. It coils more and more, and also will start touching in different places because it's three-dimensional now, so it'll start to bend like this, and it's hard to see the 3D nature in this video, but that's the important part, is that it's 3D, and then our quaternary is so multiple 3D pictures of tertiary coming together to form one big protein. So that was the importance of that video. I just wanted to show you guys the way things can look. One second, I gotta get back to my present mode. There we are. Okay, so proteins, their main functions are to catalyze reactions, which the idea of catalyzing reactions means that we will make the reactions easier to occur, more likely to occur, and will happen without being consumed by the reaction. They can transport molecules, which is very important. They can transmit messages from cell to cell. Oh, here I defined catalyze. So it's a process by which a substance speeds up a reaction without being used. But our main functions, proteins can do a lot. They're mainly, though, used for a lot of reactions, transportation, and to transmit messages. So I'm going to stop there. In our next video, we will start by talking about lipids.